The overall goal of this procedure is to produce fully human monoclonal antibodies against a desired antigen, starting from human B lymphocytes circulating in the blood.
This protocol allows the generation of monoclonal antibodies that can discriminate between highly related antigens. For some applications, such as immunotherapy, precise targeting of an antigen can be indispensable.
The advantages of this technique are that it can be performed starting from the blood of healthy or immunized donors and allows the isolation of even rare antigen-binding B cells. While this method is described starting from human samples, it can also be applied to animals that can be pre-immunized.
Visual demonstration of this method is critical as the procedure relies on a quite complex multiparametric gating strategy of discriminative B lymphocytes.
The procedure will be performed by Mélinda. She is an assistant engineer from our laboratory.
First, pipette 35 milliliters of diluted cell suspension on 15 milliliters of density gradient in a 50 milliliter conical tube. Then, place the conical tube in a swinging bucket rotor with brakes off and centrifuge at 1290 times gravity for 25 minutes at room temperature. Next, at the interface of the density gradient medium and plasma layer, carefully aspirate the mononuclear cell layer and transfer to a new 50 milliliter conical tube.